Welcome to Value-Based Voices, a podcast from the Association for Value-Based Cancer Care. Each episode dives into the shifting terrain of cancer care in the United States, exploring what value means in today's clinical, policy, and patient-centered environments. Our mission is to spark informed dialogue, promote transparency, and equip every stakeholder, from providers to payers to patients, with the insights they need to navigate cancer care with clarity, confidence, and purpose. So let's get into it. First of all, I'll introduce myself. My name is James Hamrick. I'm a medical oncologist. I'll be your host today. I currently serve as the chair of the Precision Oncology Alliance at Caris Life Sciences, a molecular diagnostics an AI company. And today we're going to be talking about with John Fox, who will introduce himself in just a minute, about some of the incredible progress that's been made in understanding tumor biology and optimizing cancer care in the diagnostic space, and some of the hurdles, challenges, and opportunities that we've got in that space. So John, welcome this morning, and I'll let you go ahead and introduce yourself.
Thanks, James. John Fox. I'm a pediatrician and epidemiologist. I've spent 20 plus years working for health plans developing medical policies, but for the last 4 years, I've been the senior medical director in market access for Illumina, which is the world's largest manufacturer of sequencing equipment.
Thank you, John. And that's really impressive. You know, I'm old enough in my career to have witnessed, I remember the Human Genome Project and the incredible effort that took from so many in the scientific community to sequence the first genome. It took many years and many dollars. Yeah. And it's just amazing to see what we can do now. Maybe walk us through a little bit about what's possible now in terms of understanding tumor biology with sequencing of DNA and RNA.
Okay, so I'm not an oncologist, I'm just a pediatrician, so I'm not sure I'm going to hit the mark. But, you know, it's 2003 when we first sequenced the human genome, and I think the publicly reported cost of that was $100 million. Fast forward 22 years.
And that's for one genome.
That's for one genome, yeah. And fast forward now, and we can sequence 200 genomes in under 48 hours at the cost of about, well, I probably shouldn't say the cost, but it's significantly less than $100,000. 3 orders, 4 orders of magnitude less than that first genome.
Right. Yeah. And I think it's fascinating. You know, part of the learnings I've had on my journey is there's the sequencing, which is sort of a science fiction miracle to be able to do it at the scale that
3.2 billion base pairs.
Yeah, exactly. And just when you think about what it is, you know, it's really it's the incredible blueprint that can tell us so much about cancer and other diseases as well. And I really believe we're at the beginning of that. The other piece, though, is when you sequence, you have a sequence of A, C's, T's, and G's. And then what many people don't totally understand is then there's the interpretation and analysis of that. And so what we call the bioinformatics. And that's really a part that is very nuanced and still very much, you know, tons of opportunity, but also tons of challenges there.
Yeah. And you pointed out in your introduction that Caris is an AI company. So is Illumina. We're using AI to simplify the interpretation and leave the final touches to the laboratory physicians or others. But that has really simplified the process and speeded the process of getting information into the hands of clinicians.
Yep. So with my clinical hat on, you know, during my career, I was able to witness the first sort of rationally designed targeted therapy, what many people would say, a drug called imatinib for chronic myeloid leukemia. I had a patient in my fellows clinic that was alive and being treated by James Hamrick as a lowly intern because of this amazing drug. I didn't take any credit for it. Yeah. Someone else had actually treated the patient, but it was amazing to see. And now we see this explosion of innovation. But let's talk a little bit about the, as with any time we have a major leap forward in the science, whether it's diagnostics, therapeutics, sometimes not everyone can benefit from that right away. You've done a lot of work in the field of appropriate use of testing. I'd love for you to sort of sketch out for listeners what we mean by that. You know, how when this technology lands, it doesn't necessarily get deployed and implemented everywhere.
Yeah. Well, first of all, it's safe to say that with any new technology, our ability to implement the technology lags behind the ability to execute the technology, meaning the process today of running the whole genome sequence, whole transcriptome sequence is pretty much cookie cutter. The process of interpreting adds a little complexity, but the process of implementing that into a clinical practice and making sure that patients benefit from that technology in terms of improving their health outcomes is widely varied across the country. We know today, for example, in non-small cell lung cancer, that about 50% of patients who should be tested aren't. The consequences of that are profound, but misunderstood and not understood at all, for that matter. For example, there are about 5 different papers that have been published that suggest that patients who are appropriate for testing who don't get it and don't get targeted therapy have a survival deficit of between 8 and 14 months.
Yeah.
If we had a new drug that improved life expectancy by that amount, everybody would be on it.
Yeah.
And the fact is that we just don't have the systems today to ensure that physicians order the test, get the test results, interpret the test results, and get patients on the right therapy. That's changing, but changing slowly. It's tragic that we have that ability, and yet not every patient has access to those test results.
Yeah, and you see that play out. In my job, I spend a lot of time traveling around the country and seeing different settings where cancer care happens in large health systems and academic centers and community practices. And what you say absolutely rings true. In your experience, what do you see as some of the barriers? Someone who hasn't spent time like the two of us have in the world of cancer care and sequencing might think it can be sort of plug and play, but maybe what are some of the cultural or systemic barriers to...
In 2022, Helen Sadek, Gerald Pritchard from the Personalized Medicine Coalition published a paper that outlined in non-small cell lung cancer a lot of the barriers to implementing precision medicine approach, including, not ordering the test, not having the sample set sent, and others, not getting the test results in time, not getting the test results at all. So there are a lot of those barriers, but certainly there are also reimbursement barriers. As a practicing oncologist, you're very concerned about financial toxicity, and sometimes payers don't cover those tests, and they can be $3,000, $4,000 to help inform what the right therapy is for that patient. And if physicians are unsure if the test is going to get reimbursed, they might order a test that they know will get reimbursed.
Right.
And ironically, a lot of insurers today cover less than 50 gene panels, which is another story for another day. But those 50 gene panels don't capture all the biomarkers that are recommended by the National Comprehensive Cancer Network to assess what therapy is appropriate for a patient. The fundamental question, I think, for you and any oncologist is what are the appropriate therapies or therapeutic options for my patients? And not testing for all the relevant biomarkers leaves you in an information void.
Yeah, I mean, it's absolutely true. So, you often find yourself in a position where you really want to, from a scientific standpoint, of course, you want to find out what are the actionable alterations. Is there another line of therapy that might extend the life of this patient or even save this patient's life, even if they were static? So for a disease like non-small cell lung cancer, there are 7 to 12 biomarkers, and you can quibble about what the science is to back them, but guidelines supported. They move very quickly. I know at Caris and sort of in my practice, when I was seeing patients, there was a between having the tissue sample or the blood sample available, usually tissue because that's the hard one to get. You really want to be as comprehensive as you can from the outset, because it tells you, it gives you the best picture of what's going to happen with the patient. At the same time, I understand the reasoning behind saying, well, if you're a payer, why would you want to pay for hundreds of alterations when there are only 8 that are actually on the NCCN guidelines? So there's an operational conundrum as well. It would be one thing if you could walk in and press a button and get the one next biomarker that's most valuable automatically delivered for a fixed price all the time. It's just, that's not the way the world works.
It's not. And in the FDA approval process for companion diagnostics, you can add additional biomarkers if they were already in, that gene was already in the test.
Right.
But for example, if you didn't have RNA in your test, you can't add that to your DNA-based test without voiding the companion diagnostic approval. And the challenge with diagnostic tests, as you point out, you simply can't add an additional biomarker. You have to go through a whole analytic and clinical validation again. And so the notion of a large panel is that you're going to future-proof your test in the sense that things that might come in the future are already built into the test.
Yeah, I mean that is sort of the I know at Caris that's the approach that we've taken
Yeah.
is to be comprehensive from the outset, whole exome on the DNA side, whole transcriptome on the RNA side, and even
Yeah.
moving towards whole genome. And we're not the only ones, but I think that is this sort of philosophical approach of future proofing and saying, look, it's better to do the entire run because you may find things that even for matching for a clinical trial,
Yeah.
or now that patients are living years and years with these diseases, thanks to advancements in diagnostics and advancements in therapy, several new biomarkers may come about.
Yeah.
And so, and then of course there's the other piece of having informed patients who have a decent understanding or the best that they can of what's going on with their tumor.
Yeah. You brought up the topic of clinical trials. One of my jobs is to work with payers to convince them of the value of,
Right.
of comprehensive testing. And one of the things that we point out is first that if you want to know all the treatment options for many cancers now, a clinical trial is a first or second line option. Yeah. And if you don't test for those, you don't know. And if you wait to test until the patient's progressed, the likelihood of enrolling in that trial is very low because the patient wants to get treatment, and we can't wait 6 to 8 weeks to find out what the patient is eligible for, get them vetted and enrolled. And so I think the notion of a comprehensive profile allows for identification of both FDA-approved drugs and identification of patients for clinical trials.
Yeah, I mean, I don't know the exact number, but I've seen reports of something like 60+% of all the oncology trials are biomarker guided in some way. And any medical oncologist out there that has seen patients who exhaust standard therapy. So this is missing all the first, second line therapies if they haven't done the testing. But if you exhaust that, your patient still is in good enough shape with good enough performance status to be treated, and you send them, for instance, to a phase 1 center to see what's available. The first thing they do is say we need a comprehensive panel because it's exactly what you said. Yeah. We can't even begin to understand what the trial opportunities are without that because so many of them are biomarker driven.
Right. Yeah.
So you mentioned value though. It's a challenge I think many of us have the belief that by understanding as much as we reasonably can related to tumor biology as early as we can, we're going to be able to make better decisions, which ultimately, over the course of a patient's cancer journey, will drive better value, keeping them out of the hospital due to less toxicity, fewer changes, keeping them on a therapy longer, even if it may be an expensive one, probably better if it's the right one and they do well and they have fewer side effects. What have you seen in terms of efforts to actually demonstrate the value of doing the appropriate test at the right time in the patient's disease? I know that's hard to measure.
Yeah. Well, the studies that I cited earlier, that patients who are on the wrong therapy have a survival deficit. There's also not only a clinical harm associated with that, there's a financial waste associated with spending. If you look at a PD-L1 based triplet therapy, that's $30,000 a month. I have somebody on that for 6 months before I recognize that they're not responding. I've just spent $200,000 on wrong therapy. Yeah. And so there is a value in ensuring that patients are on the right therapy. I think the irony of this to me is that we're quibbling with insurance companies about spending $800, $1,000 on a diagnostic test versus $3,000 when the real expense is in the therapy. Right. And if we want value, we should ensure that patients are on optimal therapy and not simply rely on a suboptimal test to help us identify right therapy.
Yeah, no, that makes a ton of sense. And the other, so very interesting in the case of molecular residual or minimal residual disease, whatever the flavor of the day is there. This idea that actually doing the right test at the right time can lead to de-escalations in therapy. So there are patients out there, particularly, or it's an easy example perhaps in the early stage setting, in the curative setting, who perhaps can avoid some of the adjuvant therapies and all of the toxicity and all of the cost associated with that if they're found to be, for instance, negative on an MRD test.
Yeah.
The other side of that coin is the patients who might be positive might need an escalation of therapy at some point. So what do you see in terms of adoption of that type of logic that, maybe historically we've said find the one biomarker that allows
Yeah,
you
yeah,
to then put them on the right therapy, but it maybe allows them to stay off of the wrong therapy.
right. Yeah, I think the kind of the prototype for this was the Oncotype DX test in breast that came to market, I don't know, 15, 20 years ago,
Right.
in which we could identify patients who didn't benefit from chemotherapy and save not only the cost, but the toxicities and the harms associated with that. And I think that's the promise, the potential, and ultimately the promise of MRD incorporation into the treatment algorithm to identify those patients who don't need therapy.
Right.
And the interesting thing about the Oncotype DX test is that there was pretty rapid payer adoption of that because the evidence supported it, even in the retrospective trial
Right.
upon which the test was approved, because it reduced cost. And I think if we can, it's unfortunate, but I think it's true that payers will gravitate and lower their evidentiary standards if they think that something is going to reduce the total
Right.
cost
Yeah.
of care. As a former health plan medical director of 20 plus years, there are very few things that I could say are going to not only improve outcomes, but also reduce the total cost of care. And so I think there has been widespread adoption of MRD testing in hematologic malignancies.
Right.
Certainly, MolDX and Medicare, MolDX has adopted coverage for a lot of solid tissue tumors, and some health plans have as well. But I think, you were at ESMO the last week, and there were a number of studies published showing the value of MRD,
Really exciting.
including in muscle invasive bladder cancer. So I think in the next 5 years, we'll see a proliferation of studies that show the benefit of that. And I anticipate that payers will adopt those, especially in those settings where it reduces the toxicities associated with unnecessary treatment.
Yeah, yeah. And you talked about Oncotype DX. That represents another interesting element of comprehensive testing. So that is a signature,
Right.
which means it's not just one gene. We've often thought of one gene that has
One
an
drug,
alteration,
right.
one drug. Oncotype DX has been around 20 years in practice, or 15 years, something like that. And it is actually a signature, which is a combination of different genes with alterations. Now that with comprehensive testing, you can build signatures, and we see this, and lots of the smartest investigators in the cancer space are thinking about how can we create gene signatures that will help us with important clinical decisions. What's the optimal first-line therapy in metastatic colon cancer? FOLFOX, FOLFIRI, something else. We're going to see these more and more. That's another sort of another proof point of the value of being more comprehensive. If you're looking at the whole exome, whole genome, and transcriptome, it gives you a better opportunity, especially now that we have artificial intelligence and the ability to crunch large data sets to find signals among combinations of different gene alterations that can really.
And those are
transform
already here.
patients.
We've got homologous recombination deficiency, MSI, TMB.
TMB,
Right.
exactly. And refining some of those is that, there's some interesting work going on about refining TMB. It probably not one size fits all. So very, very interesting stuff.
And I think that on the payment side, the challenge is that we're trying to explain something that's fairly complex and put it in terms that payers can understand. I was a former payer. I was a pediatrician. I could spell DNA, so I was qualified to write policy. But that's a challenge in trying to translate the science into terms that business people can understand in terms of writing policy. But I think that's just one element. We've got the technology. We've got the ability to interpret the genome. We're working on coverage, but it still comes back to, does the physician have the tools to know when to order a test and what the right test to order is? As an example, we're working with EMR vendors and others to help a physician answer 3 questions. Number one, if I've got a patient with cancer, what are the NCCN recommended biomarkers for that cancer type? Number 2, for this patient's insurer, which of the tests available on the market actually capture all those relevant NCCN biomarkers? And number 3, using the digitized policy for that payer, would this patient meet the payer coverage criteria? And physicians have said, if I had all that information, that would be the cat's meow, because that's my biggest barrier to ordering a test. So I think fundamentally, we have all the pieces, but we're not Battlestar ready, because we can't consistently ensure that patients get the right test at the right time in the right therapy
Yeah.
at the right time.
Well, I love the practical thinking. So, busy oncologists have workflows that are working for them. And when you think about how do I integrate this into the workflow of a busy oncologist, the sort of simplify, first of all, asking them exactly what they need in order to be able to make it easier to do the right thing. And then the simplified version of getting it into their EHR workflow sounds like a solution with a lot of legs.
It was interesting this morning, right before our conversation, Seema Verma used to be at CMS and is now at Oracle, was talking about the development of their EMR, and they have the ability to do all those things that I just said. And I think EMRs are huge investments, as you can appreciate, but to the extent that it improves patient care and improves patient outcomes, and I think about the quadruple aim, that the last element in the quadruple aim is improving the physician and healthcare provider experience. If all those things are possible with the new AI-enabled EMRs, I think there will be adoption, even if it's an extraordinarily expensive adoption or conversion.
It will be. But nothing that's worth it is.
And hearing that kind of thing today at the AVBCC just gives me hope that the application of other technologies will help enable technologies like genome sequencing to enable kind of a personalized and precision medicine approach.
Yeah, it's very exciting, and a very, very exciting way to think about making sure that the benefits of all this great technology reaches everyone and doesn't actually drive a larger inequity in care. So John, thank you so much for sharing your expertise.
You're welcome. This has been a great conversation.
I enjoyed it. This is the kind of meeting at AVBCC you hear from thought leaders like Seema Verma. You get inspired, and then you carry on and try to do something a little different the next week. So thank you so much. This wraps up this episode of Value-Based Voices, brought to you by the Association for Value-Based Cancer Care. A heartfelt thank you to John Fox for sharing his insights and expertise, and to you, our listeners, for joining the conversation. If you found today's discussion thought-provoking, be sure to subscribe to Value-Based Voices wherever you get your podcasts. Stay tuned for more episodes that spotlight the voices that are shaping oncology.